Hello everyone. This class is about molecular cloning. In the last class, you learned about how to use PCR reaction to amplify your DNA fragments, including your backbone, which means plasmid, and your gene of insert. And today, molecular cloning tells you how to assemble them into the real machine. What molecular cloning is about is just to isolate the DNA fragments first, which is the backbone, the plasmid you want. Second is your gene of interest into uh, into one product called the recombinant clone. After we introduce the recombinant clone into your biological environment for amplification, organisms containing these copies of the clone DNA segments are selected by multiple selection method that we mentioned, like antibiotic selection or selection markers with different properties. Afterwards, the covalent clone is amplified, and then we can get multiple identical DNA plasmids, which is called the DNA clone. Before we get into how we actually assemble our DNA fragments together, let's first review the fundamental of them, which is base pairing. All of the assemble methods that we're going to mention later is based on the very basic Watson-Crick DNA base pairing, which means that the adenine is always paired with thymine, A and T, and the guanine is always paired with cytosine, C and G. This kind of complementary base pairing is most stable, was the most stable in the thermodynamics. But however, we have some other method of DNA base pairing, especially when mutation occurs in the DNA. When the cytosine mutated with methylation, a double base pairing will happen, which means G pairs with T. After reviewing about knowledges of DNA base pairing, let's first get into what's called a molecular cloning and the very traditional way of molecular cloning, which is called the, the restriction ligation. First, please pause here and watch the Canvas video 1, Molecular Cloning Explained for Beginners. Welcome back. After watching the video, I believe you have some brief understanding of how restriction enzyme and restriction ligation work, and let's dig a little bit more further into it. There are a few hundreds of these kind of restriction enzymes, and you may see them with some strange name, but don't worry. They're named after the species which they are identified. For example, the most common one, EcoR1, is just discovered in E. coli. Let's further assess what is a restriction enzyme. Their full name are restriction endonuclease. Restriction means that their cut are precise and predictable. Endo means that they cut the DNA fragment inside the DNA instead of cut the DNA fragments at the end of the DNA. Nuclease means that they cut the backbone of the sugar phosphate bond. These enzymes could recognize the specific sequence and precisely make two cuts on each strand of the DNA. As you can see, they can cut with the two strand at the same side to create a blunt end with no what's called a overhang. But some enzymes could leave one single-stranded overhang to produce a sticky end. A sticky end means that when two complementary ends meet together, they can be easily ligased, annealed, or you can say connected together. So let's see how restriction enzymes creating a combined component DNA. First, your plasmid and your DNA fragments of interest were handled with restriction enzymes, which may be the same, may be different, but they create the same sticky ends. And after these process, the DNA fragments could be annealed with their complementary sticky ends to form a recombinant DNA molecule. And such recombinant plasmid could be translocated into your host cell. Different enzymes which results in complementary sticky ends could also be used. And enzymes forming blunt end doesn't necessarily need to be has any complementary sticky ends, and any blunt end could be ligased together with specific ligase. Ligated together with specific ligase. The next novel approach of assemble your DNA fragments is Gibson assembly. This approach doesn't limit it with sticky ends or complementary ends, but you have to carefully design your overhang. Now please pause here and watch the Canvas video 2, a detailed look at Gibson assembly. Welcome back, and we believe you have a further understanding of how Gibson assembly works. Next week, we will teach you practically about how to design overhang for assemble your gene of interest into your designated plasmid. Thank you.